Welcome to the Sand Science Experience podcast, where we explore the latest and most exciting papers and preprints in science. Before we jump into today's deep dive, please take a moment to like, subscribe, and share this information with your friends and family. Your support really helps the Sand Science Experience grow. Today, we're embarking on a really fascinating deep dive. It takes us right to the heart of cellular life, looking at how, broken machinery affects the power grid inside our cells. We are digging into research that explores how deficits in ribosomal proteins, these RPs, the core components of your cell's protein factories, don't just slow things down. They actually directly and, interestingly, differentially impact mitochondrial function and your overall cellular energy balance.
Yeah, it's quite an incredible insight into what seems to be a conserved survival mechanism across species. So our mission for you today is to unpack the findings from this specific research. They used C. elegans, those tiny nematode worms, and also human lymphoblast cells. And we're focusing specifically on this genetic vulnerability called haploinsufficiency, which, sounds complex, but it just means having lost a single copy of certain ribosomal protein genes. And we want to understand how that single loss can completely throw off the delicate cellular energy equilibrium. This has massive implications for human diseases like Diamond-Blackfan anemia, DBA, and maybe even cancer susceptibility.
Okay, let's unpack that interplay then, starting with the basics. These RPs, ribosomal proteins, they're essential for making proteins, right?
Absolutely essential. They form the ribosome itself.
And haploinsufficiency means the cell is basically working with only one good instruction manual instead of two for a specific part, like running on 50% capacity for that particular piece.
Exactly. Half the normal gene dose. And that 50% isn't just a minor hiccup. It's actually the root cause of some significant human health problems. Haploinsufficiency, or sometimes specific point mutations in these RP genes, cause a whole group of disorders we call Ribosomopathies. DBA is sort of the classic example. People with DBA often have serious issues with blood cell production. But what's really startling is how broad the relevance is. Similar losses in these RP genes are found surprisingly often in something like 40% of human tumors. So it's not just about development, it links ribosome stability directly to cancer.
Wow, okay. So the link to energy, building ribosomes must take a lot of energy, I imagine.
A huge amount. It's one of the most energetically demanding processes in the cell. So ribosome production is inherently tied to mitochondrial function, the cell's power plants, and keeping that energy balance, the equilibrium.
And you mentioned a clinical parallel.
Right. This is what really caught the researchers' attention. The symptoms of DBA look strikingly similar in some ways to another condition called Pearson syndrome. Pearson syndrome is caused by mitochondrial DNA loss. So seeing that overlap suggested, maybe problems in the ribosome factory and problems in the mitochondrial power plant share some kind of deep, fundamental vulnerability.
A shared weakness. That makes sense. So they use C. elegans to investigate this.
Exactly. It's a great model for this kind of work. They looked at worm strains that were haploinsufficient for 4 specific RP genes: RPS10, RPL5, RPL33, and RPS23. These cover parts of both the small and the large ribosomal subunits.
Okay, and here's where it got surprising, you said. Usually if you break a core part of the cell, you'd expect it to be weaker or less resilient.
You would think so, right. But these RP haploinsufficient worms showed unexpected resilience. Every single one of those strains was significantly more resistant to acute oxidative stress. They tested this using paraquat, which is a pretty harsh oxidant.
Huh. So why would a partially broken cell suddenly be tougher against poison?
Well, it suggests the cell's already kind of on high alert. It's switched into a defensive mode. This enhanced resistance seems to come from activating a key stress response pathway. It involves a transcription factor called SKN1 in worms. That's the equivalent of NRF2 in mammals, which is quite well known. When the ribosome is under stress from being incomplete, it signals to activate SKN1. And SKN1 then boosts the production of detoxification genes, especially these glutathione transferases, the CLIS2 genes.
Ah, okay. So it's like the cell is diverting resources, building up the defenses instead of focusing on, say, growth.
Precisely. It's a classic resource reallocation strategy. They're fortifying the walls, so to speak.
But that kind of strategy usually comes with a cost, doesn't it?
It does, and the paper notes a significant trade-off. While these worms are stress-resistant, the RP mutants generally show pretty clear developmental delays. The cell is putting so much energy into survival and defense that normal growth kind of takes a back seat. It's literally choosing defense over development.
Okay, so this sets the stage for the really specific finding you mentioned earlier, because not all these RP losses affected the energy production equally.
Exactly. Even though all 4 mutants activated that general SKN1 stress response, Only the loss of one specific RP, RPS10, which is part of the small ribosomal subunit. Only that one specifically seemed to compromise mitochondrial function in a major way.
Right. So the big question is, why RPS10? What was different about the RPS10 mutants?
Well, the functional defects they saw were pretty stark and directly related to energy production. The RPS10 worms showed significant drops in mitochondrial membrane potential.
And that potential is critical, right? It's the driving force for ATP production.
Absolutely critical. If that gradient collapses, the power plant basically shuts down, and they saw the consequences. These mutants had elevated ADP-TP ratios. Think of ATP as the charged battery and ADP as the used one. A high ratio means too many used batteries, the cell can't recharge them fast enough, clear sign of disrupted energy balance.
Okay, so lower potential, messed up energy balance. Did they consume less oxygen too?
They did. Reduced oxygen consumption rates. It all pointed towards the mitochondrial engines just not firing properly. A very clear picture of energy failure tied specifically to losing one copy of RPS10.
And you mentioned this wasn't just a worm thing. This effect is conserved.
Yes, and that's really important. They confirmed it in human cells. They used RNA interference to reduce RPS10 levels by about 50% in human K562 leukemia cells, and the results mirrored the worm findings. Decreased mitochondrial activity, increased ADP-T ratios. So it strongly suggests this is a fundamental conserved pathway linking RPS10 to mitochondrial health.
Okay, so if the effect is conserved, what's the mechanism? How does the cell actually manage protein production when these parts are missing? You mentioned something about translational buffering.
Right. This is where they used a technique called Ribosec to get a deeper look.
Ribosec that basically tells you what proteins are actually being made efficiently, not just what RNA exists.
Exactly. It's a snapshot of ribosome activity. And what they found was fascinating. The cell coordinates things very carefully. When an RP is lost, the cell first tries to compensate by making more RNA for all ribosomal proteins. It prints more blueprints. But translating all of those would be wasteful. So at the same time, it actively reduces the translation efficiency, the TE, of those very same ribosomal transcripts.
So it makes more instructions, but slows down the actual assembly line for those parts.
Kind of, yeah. It's like having more raw materials delivered, the RNA, but controlling the flow through the final assembly stage, low TE. This translational buffering helps maintain the overall number of functional ribosomes without wasting too much energy. It's quite sophisticated.
Okay, that makes sense for ribosome parts. How does this buffering connect back to the mitochondria and the electron transport chain, the ETC, the energy machinery itself?
Good question. So the ETC components, the proteins that actually perform OXPHOS, pronounced OXPHOS, oxidative phosphorylation, they're made from instructions in both the nucleus and the mitochondria's own DNA.
Right, two different production lines feeding into one machine.
Exactly. And here's the differential effect. They found that the ETC parts encoded by nuclear genes made by those main cytoplasmic ribosomes, their translation efficiency wasn't really affected. However, for the ETC components encoded by the mitochondrial DNA things, like key parts of complex 1, their translation efficiency was significantly reduced in several of the RP mutants, including RPL5 and RPL33.
Wait, so a problem in the main ribosome factory, even one that's being buffered, somehow slows down protein production inside the mitochondria.
It appears so. It suggests that maybe defective cytoplasmic ribosomes, even indirectly, impair the translation happening inside the mitochondria. This could lead to a shortage of crucial internal ETC components, hobbling the power plant. And the coordination seems incredibly tight. They looked in human cells too and found a significant correlation, a covariation, between how ribosomal genes and mitochondrial membrane genes are expressed, both at the RNA level and the translation efficiency level. It highlights this really intricate regulatory network.
Okay, let's tie this back to the stress pathways before we wrap up. You mentioned SKN1-NRF2 earlier. The study also looked at the integrated stress response, the ISR, and a factor called GCN1.
Yes, GCN1 is a key player in the ISR, often involved in regulating protein synthesis during stress. We already know that just disrupting GCN1 on its own can lead to reduced mitochondrial function in worms. So the researchers looked at what happens if you disrupt GCN1 in the worms that also have the RPS10 defect.
And what did that interaction show?
This is where it gets a bit counterintuitive, but really interesting. In worms lacking functional GCN1, the negative impact of losing RPS10 on mitochondrial function was actually lessened. It was minimized.
So getting rid of a stress regulator helped in the context of the RPS10 defect.
It seems so. It suggests that GCN1 is somehow required for RPS10 loss to exert its full negative effect on the mitochondria. The RPS10 problem might not hit the mitochondria directly, but perhaps acts through this GCN1 pathway. It points to a regulatory chain linking the ribosome, the ISR, and mitochondrial energy production.
That adds another layer of complexity. And finally, did they actually look at the mitochondria themselves? What did they look like?
They did, and they found that all the RP haploinsufficient mutants seemed to struggle with maintaining healthy mitochondrial structure. They showed more signs of mitochondrial fragmentation. Instead of a nice interconnected network, the mitochondria looked more like small, isolated, round units. This fragmentation was especially noticeable when the worms were put under a bit of temperature stress, around 23 degrees Celsius. It suggests a general fragility and inability to maintain the network under pressure.
Right, so bringing this all together, the key takeaway seems to be that ribosomal proteins, and RPS10 in particular, aren't just passive building blocks. They're acting almost like sensors, deeply connected to the cell's power grid. And losing just one copy of RPS10 sets off this conserved stress response, but ultimately disrupts mitochondrial function and energy balance, likely through this careful yet ultimately insufficient translational control of ETC components.
Absolutely. And it leaves us with a really provocative question, a bit of a paradox, actually. Given that most of the RP mutants showed similar translational buffer effects, seemingly throttling mitochondrial translation in a similar way, why did only the loss of RPS10 cause that major functional collapse in the mitochondria? Does RPS10 have some unique, maybe non-canonical role we don't know about, perhaps directly interacting with the mitochondria? Or are the other RP mutants just better at activating some downstream compensatory pathways that manage to buffer the functional impact, pathways that RPS10 loss fails to trigger effectively? It really suggests RPS10 might hold a specific crucial key in this ribosome-mitochondria communication network that we still need to figure out.
A fascinating puzzle with clear implications for understanding diseases like DBA and maybe even cancer metabolism. It's definitely something for future research to explore. Thank you for walking us through that intricate cellular story, and thank you all for joining us on this deep dive. Remember to like and subscribe to the Sand Science Experience to keep exploring the latest and most exciting papers in science.